bases and salts. Introduction. So before diving deep into the lesson, just recall some questions and we'll try to find the answers of these questions as we go throughout the lesson. Okay, so here it is. Why are lemons sour? Why are soaps slippery? Are all acids corrosive? Are amino acids really acids? And if yes, how can they be within our bodies? What about vitamin C? Is it an acid or a base? What about all those pH balanced shampoo we find at supermarkets? How do you exactly go about treating a bee sting? What has happened to Taj Mahal? How had it become yellow? Are cola really good for teeth? What exactly are we tasting when we taste something bitter, sour or sweet? So you'll find the answer to all these questions by the end of this lesson. So let's dive deep into the lesson. Here it is. So basically matter is divided into three types: acids, bases and salts. So it is a human tendency to classify everything, right? We humans classify matter into solids, liquids, gases, into acid, bases, salts and so on. Classification is better for us to remember things. So let's talk about acid, bases and salts here. So acids are sour in taste and change the color of blue litmus to red. Bases are bitter and change the color of red litmus to blue. Salts are substances which are produced by the reaction of an acid with a base. So when acid and base react with each other, it forms salt. Now, acid turns blue litmus to red, while base turns red litmus to blue. So here, what is litmus? Do you know? Litmus is an indicator which helps us understand whether some substance is acidic, basic or neutral. Now, you can remember this conversions of color by like this, watch out for base. Base turns red litmus to blue, right? So base, blue, B, right? So you just remember that base turns red litmus to blue, and the opposite will be for the acids, then which is blue litmus to red. Okay, so this is how you should remember, or I'll suggest you to remember. If you have any better idea, you can go for it. Litmus and turmeric are natural indicators. So what indicators are meant here is that it will help us know whether a particular substance is acidic, basic or neutral. Now litmus we know, right? Litmus paper are being used into laboratories. But what about turmeric? Do you have any idea how these turmeric indicators are used, or how do they give us the solution whether any substance is acid, base or neutral? Have you noticed that a stain of curry on a white cloth becomes reddish brown when soap, which is basic in nature, is scrubbed on it? So a stain of curry on white cloth becomes reddish brown when soap is rubbed, because the curry contains turmeric, which is a natural indicator, whereas soap is basic in nature. When turmeric reacts with soap, it turns into reddish brown. So you can actually perform this. Okay, don't stain curry on white cloth, of course. But you can just take a pinch of turmeric powder and put it over the soap or any kind of soap. What you will see is that the turmeric powder turns red. This is because soap is basic in nature and thus it is slippery and bitter as well. So soap is basic in nature, and turmeric when turmeric is added to any base, it turns into reddish brown color. So you can just perform this activity in this way. When it is washed in water, it turns yellow, because when turmeric comes in contact with water, which is neutral, it becomes very dilute and the reaction stops. You can also use synthetic indicators such as methyl orange and phenolphthalein to test for acids and base. So you can say that indicators are divided into natural indicators and synthetic indicators. Natural indicators are those which we have already found ready-made from the nature, while synthetic are those which we made from laboratories. Methyl orange is one of the synthetic indicator. Methyl orange is basically orange in color, and it gives a color change in presence of acids, bases and neutral as well. Also, phenolphthalein is something like this. Like when you to get results, the phenolphthalein changes its color to pink. Okay, so we'll learn about this in further modules. For now, just remember these names: methyl orange, phenolphthalein, etc. Now, litmus solution is a purple dye which is extracted from lichen, a plant belonging to division Thallophyta, and is commonly used as an indicator. It is absolutely okay if you don't remember what division Thallophyta is, or if you don't know what it is. We will learn about this in further modules. But for now, just remember that litmus solution is a purple dye and it is extracted from lichen. This is an important fill in the blank question, so just remember it. Now, when the litmus solution is neither acidic nor basic, its color is purple. There are many other natural materials such as red cabbage leaves, turmeric, colored petals of some flowers such as hydrangea, petunia and geranium, which indicate the presence of acid or base into the solution. So all these names which I took now, red cabbage leaves, turmeric, colored petals of those flowers, are nothing but natural indicators. They are ready-made, available for us to use by the nature itself, while synthetic ones are phenolphthalein, methyl orange, etc. So this was the introductory module. We will learn about acids, base and salt much deeper in further modules. So thank you.